We are excited to have Dr. Valerie Pence here speaking tonight on saving all plants, technologies to extend ex situ conservation of all plant species. Dr. Valerie Pence is the Director of Plant Research at the Center for Conservation and Research of Endangered Wildlife based at the Cincinnati Zoo and Botanical Garden. That's a position she's held since 1988. She's also an adjunct research professor in the Department of Biological Sciences at the University of Cincinnati. Dr. Pence received her BA in biology, specializing in botany at Mount Holyoke College here in Massachusetts. And from there, she went to Northwestern University in Illinois, where she earned a master's degree and PhD in plant physiology. Dr. Pence has numerous publications on the topics of plant conservation, plant physiology, seed biology, embryo biology, in journals like Biological Conservation, Plants, People, Planet, Hort Technology, and the American Fern Journal. She's also received the Center for Plant Conservation Star Award in 2010 and the Garden Club of America's Conservation Commendation Award. So we're very excited to have Dr. Valerie Pence here this evening. Most recently, just last month, sort of hot off the press, a new review paper came out that Dr. Pence was the lead author on, focused on the topic of exceptional plant species and some of the challenges of preserving them ex situ. And tonight's talk will be based in part on this recent publication. And I wanted to sort of flag also for folks in the organization, if you notice the eighth author on this paper is our very own association manager, Sarah Held-Wallace, who was a collaborator on this project. She worked with Dr. Pence when she was a master's student at Longwood Gardens in the University of Delaware. Dr. Pence served on Sarah's master's thesis committee. And the New England Botanical Club is indicated here amongst the addresses of the authors, given Sarah's position with our organization. So great connections here. We're very excited to hear from Dr. Pence this evening on the topic of conserving exceptional species, so saving all plants, technologies to extend ex situ conservation of all plant species. So with that, I'll turn it over to Dr. Pence. Thank you so much for joining us tonight from Cincinnati. It's great to have you here, and we're all excited to hear about your work.
Thank you very much, Jesse, for that nice introduction. And it's really a pleasure to be with you tonight. I've heard about your society for many years, and it's just great to be able to be with you. And I appreciate being invited. Thank you so much. Well, as Jesse said, I work at the Cincinnati Zoo and Botanical Garden at the Center for Conservation and Research of Endangered Wildlife, or CREW. And we are the research program of the Cincinnati Zoo and Botanical Garden. We are sort of off the beaten path on the edge of the campus at the moment, but we have both an animal and a plant division there. And one of the main areas of focus of our building and our projects is our ex situ collections that are housed in our cryobiobank. And it does house animal samples. Those are animal gametes of animals that are of particular interest to the zoo community, gorillas and tigers and cats and things like that. But within the cryobiobank, we also have our frozen garden, and we have a number of kinds of tissues there that I'll be talking about more throughout this talk. So I just wanted to sort of set that scene that we are going to focus primarily on ex situ conservation tonight. And that is the focus of most of the work at CREW. Just so we're all on the same page, many of you probably know the word situ comes from the Latin meaning site. So in situ conservation is the kind of conservation that is directed at preserving plants in their natural habitat, or plants and animals, in-site conservation, whereas ex situ conservation is off-site conservation or away from the natural habitat. And there are a number of ways that this can be accomplished, and I'll be mentioning all of these tonight living collections, seed banks, in vitro collections, or cryobanks. Well, why is ex situ conservation important? And I just wanted to give a couple examples to start off with. It's really an insurance policy against extinction. Many of you might be familiar with the Franklinia tree, Franklinia altamaha. It's a species of small tree that was near extinction in the late 1700s in the southeastern United States. One theory is that it may have been driven southward by the glaciers and then was left as a relic in that area of our country and was just sort of on the brink of extinction at that point. But in any event, the plant explorer John Bartram and his son William came across it during their travels in the 1760s and 70s near the Altamaha River in Georgia. Bartram collected seed and sent it to England as well as to his garden in Philadelphia. It was seen a couple times since then, but the last time it was seen was in 1803 by John Lyon, a Scottish botanist who was also exploring in the southeastern United States. And it is believed that it is now extinct because it hasn't been seen since then in the wild. However, the plant lives on in cultivation, or ex situ, descended from the plants that grew from the seed that John Bartram collected. Another example is Sophora toromiro, or the toromiro tree. This is a legume tree that was found only on the island of Rapa Nui, or Easter Island in the Pacific. Although the island was once forested, settlement by the Polynesians and Europeans removed most of the forest cover for agriculture, and the remaining trees survived only in small clumps. By the 1930s, there was only one tree of toromiro remaining on the island. It was growing in the walls of a small crater, which you can see in the center there, the Ranukau crater in Rapa Nui, where the goats and other herbivores couldn't get it. From that tree, or a few of the remaining trees before then, there were four seed collections made. Two of them were made in the early 1900s and sent to the Royal Botanic Gardens Kew and the Gothenburg Botanic Gardens in Sweden, but neither of these survived. Two other collections were made in the 1950s from that particular tree, one by Efren Voloski, a Chilean botanist, you can see on the lower left here. And that tree was grown from seed of that collection at the National Botanic Garden in Chile. The other collection was made by the explorer Thor Heyerdahl, who sent the seeds to the Gothenburg Botanic Garden in Sweden. All the known plants in cultivation today are descended from these two collections because that last remaining tree was cut down in 1960 for firewood. And the plant now grows in ex situ collections in botanic gardens around the world. And there is also an organization that's working on learning more about the genetics and finding all of the collections that are possible to collect from to try to reestablish that population of toromiro back in situ on Rapa Nui. Well, these are a couple individual cases where collecting plant material and propagating it ex situ has saved a species from extinction. But how do they fit into the bigger picture of the state of biodiversity today? The 2020 analysis from the Royal Botanic Gardens Kew on the state of the world's plants and fungi estimates that now 2 in 5 plants are threatened with extinction. The current IUCN Red List has assessed over 58,000 plant species thus far, and it has a similar percentage of those species that are either extinct, critically endangered, or vulnerable, about 40% as well. So as a result of the increasing threats to the loss of biodiversity from habitat loss, unsustainable harvesting, climate change, and the like, ex situ collections are becoming more important as a backup for these species. One type of ex situ conservation that's had a long history is that of living collections, particularly in botanical gardens. One limitation, of course, is space, and gardens often have specialized collections to maximize their space and expertise. However, most collections do not have the space for more than a few individuals of any one species, And that is the focus of a recent initiative from the Morton Arboretum and Botanic Gardens Conservation International, which is known as the Global Conservation Consortium. It's a project to coordinate collections among botanical gardens so that they can be handled as meta collections. There are a number of consortia built around different genera and groups, such as cycads, oaks, and dipterocarps, among others that you see here. However, even these represent only a small number of species compared to those that we may want to conserve. So in order to more efficiently conserve plant diversity, humans have turned to seed banking over the past century. Seed banking began as an agricultural enterprise and remains very important to maintaining the genetic diversity of crop species. Nikolai Vavilov, a Russian scientist, devoted his career to studying the genetics of crop species and started the first seed bank for crops in what later became the Vavilov Research Institute for Plant Industry in St. Petersburg. Since then, there have been many crop seed banks established around the world, and more recently, seed banks for wild and threatened species. It's been shown for many seeds that longevity increases as the temperature decreases. And so today, conventional seed banks dry seeds in drying rooms of about 15% relative humidity, and then store the seeds in freezers at about -18 or -20 degrees centigrade. That is the standard practice. You've likely heard of some of the seed banks around the world, particularly one in the upper left-hand corner of this screen, the Svalbard Global Seed Vault in Norway, which gets quite a bit of publicity occasionally. But there are a number of others. The one in the left, lower left, is the USDA's National Laboratory for Genetic Resources Preservation in Fort Collins, Colorado, which, like the Svalbard, stores primarily crop species. In the lower right-hand corner is the Millennium Seed Bank, Kew, at Wakehurst Place in the UK, which has stored so far the seeds of 1/10 of all the world's wild plant species. And in the upper right is the Germplasm Bank of Wild Species at the Kunming Institute of Botany, which is the largest seed bank in China. Seed banking is an extremely efficient method for conserving large amounts of genetic diversity in a small space, and such collections are critically important for maintaining the world's plant diversity ex situ. They take advantage of the fact that many seeds tolerate extreme drying. Excuse me. If you look at this diagram, it illustrates the relationship between water and temperature in tissues. As you move down the left, the y-axis, the temperature is decreasing. And as you move left on the x-axis, the moisture content is decreasing. So at the top of the graph, you see where most living tissues hang out most of the time. And water is in the fluid state. If you decrease the temperature, when the water contents are still that high and sort of go down the y-axis, ice will form in the seed, and it's lethal because as you know, as ice forms, it expands and breaks the cells and will kill them. However, as you move left at the top of the figure here, and if tissues become very dry, you get to a point when below about 10% moisture, where if you then decrease the temperature, you don't go through that lethal ice stage. You're in a situation where a glass has formed, and that is not going to be lethal for the tissues because the glass will not expand upon freezing. However, the tissue has to be able to survive that extreme drying. And actually, many seeds actually do survive that extreme drying, and there's no damage to them when they get to that very dry point. And then when they're put into a freezer, they're also not damaged. So that's where most of the seeds that are in seed banks fall. But there's always a but, isn't there? What about seeds that can't survive drying down to those very low moisture contents? Take acorns, for example. Most all of the oak seeds that we know of are considered desiccation sensitive. They cannot be dried beyond a certain point, down to the level of where it would be safe to be frozen. If you look on the top of this screen as well, we have vegetative tissues. We know that if you take a leaf and put it in the freezer, it's going to be killed as well. So what do we do about those kinds of tissues in trying to preserve them? They can't be conserved in traditional seed banks, and they need to be approached much differently. In fact, those were two examples of species that can't be conserved in seed banks in the conventional form. And we call these species exceptional species. The reasons for exceptionality can be organized around the steps of seed banking. And that's what you see in this diagram here. The steps are numbered. And the first step is that seeds must be available for banking. Obviously, if you don't have seeds, you can't bank them. Then in step 2, the seeds must be processed, which is the actual drying step of getting them down to a state where they can be frozen. And that's usually done, as I said, in the room that's at about 15% relative humidity. The third step is they need to be frozen and stored over a long period of time. And then they need to go through the fourth step, which is recovery. The whole point of any ex situ endeavor is to save materials for recovery and use later on. So those are the 4 steps of seed banking, but roadblocks can occur at any one of these steps, and other approaches need to be used in terms of handling them or preserving them. And that's what we're going to talk about a little bit more. I'll give you a few examples of exceptional species that may have a roadblock at these 4 steps. And we call these steps exceptionality factors if there is a challenge at that step. An example of a species with an exceptionality factor number 1 would be Hedioma todsinia. It's a federally endangered mint plant from the mountains of New Mexico, and it's found in only 2 population groupings there. But it's not producing seed. It flowers, but it does not produce seed, and so there's nothing there to bank. Quercus hinklei, or Hinckley's oak, is a critically endangered oak from the southwestern United States. As I mentioned, it's desiccation sensitive, as are all oaks, and thus the acorns can't be dried enough to freeze them. Sertander gracilis is a gesneriad from Hawaii, and it has seeds that can tolerate drying, but the seeds are freeze sensitive and don't last very long at freezing temperatures. An example of exceptionality factor 4 is Ilex paraguarensis, and it is an example of hollies. And most hollies have seeds that are deeply dormant and very difficult to germinate. They may take 2 or 3 years to germinate. And this kind of a challenge can hinder both the recovery of plants after seed banking, but also it makes it very difficult at the beginning of the seed banking process to know whether those seeds are going to be tolerant of drying or freezing, because you can't do the testing on them very easily. So all of these types of plants, all of these types of exceptionality factors pose challenges to ex situ conservation. Well, one question is, how many exceptional species are there? How big a challenge is this going to actually be? And if you go through some of the numbers, we'll just get some idea of this. We don't really know for sure an exact number. But if you take the total number of all plant species at about 305,000, there are good models for desiccation sensitive seeds, and it's predicted that about 8% of those are going to exist, or 8% of the flora is going to be desiccation sensitive in their seeds. So that gives you about 24,000. Most of these are tropical woody species, and many of them are threatened. In terms of the short-lived seeds, that would be exceptionality factor 3, the ones that don't last very long in the freezer. There may be up to 50% of all species in that group. We really don't know, but there are some papers out that suggest that high proportions of alpine species, of carnivorous plants, there has been documentation that about 30% of the Hawaiian taxa fall into this group. And then also that certain families have a tendency to be shorter lived than others, such as the Asteraceae and the Poaceae. So we really can't put a number on that, but it looks like that's going to be a large number. In terms of seeds that are deeply dormant, or the ones that have very few or no seeds for banking, we really have no predictive models at the moment for knowing how many seeds those will be, or species those will be. But if we just think about the desiccation sensitive species, and then think about threatened taxa, if that's about 40%, as I mentioned earlier, that could be at least 10,000 species that would fall into this exceptional category just from that one category alone, the exceptionality factor 2. So we really don't have a good number, but we know that it's going to be in the thousands. Well, another question that comes up is, which species are exceptional? And Jesse mentioned before that Sarah Helm-Wallace in 2015 published her thesis that included developing a list of North American threatened exceptional plants. So she sort of got the ball rolling here. That provided the basis for an even more extensive effort to create a global working list of exceptional plants. And the background for the development of that list can be found in 2 papers that were just published, as Jesse mentioned, in Biological Conservation. The list itself is now available at online at cincinnatizoo.org/EPCN, which stands for Exceptional Plant Conservation Network, which is a website that we're trying to use to develop connections between people who are working on exceptional plants, who might be interested in them, and trying to help facilitate research in that area. I should say that there are for this project, we evaluated 23,000 species. A lot of this was done sort of automatically, partially automatically, through some database work by Emily Beckman, using the seed information database from the Q, Royal Botanic Gardens Q database, and Sarah's thesis and a few other sources. Out of that, and through the process that's explained in these papers, we got about 775 species that are identified as exceptional species. As from what I just said, in the numbers of predicted exceptional species, that is a very small amount, at least only a minimum or maximum about 2 or 3% of what's predicted. So there's a lot of work to do, and we really encourage people who have any further information about other species that might be considered exceptional, or would like to look at the list and perhaps add more information about species that are already listed. We are very interested in having people do that. There's also a form online that is available for people to add their information that way. So I encourage any of you out there who might be interested to check that out. Well, how does this fit into the bigger picture of conservation? You may be familiar with the Global Strategy for Plant Conservation, which is, if you're not, it's available online. And it's a document that sets targets for various areas of plant conservation. And the target for ex situ conservation was that 75% of all threatened species should be in ex situ collections by the year 2020. While there was significant progress in that area over that decade, this target really wasn't achieved. It was achieved more in some countries than others. But it was very interesting that in 2018, there was a paper published by Weiss et al. Who did an analysis of species from the IUCN Red List and looked at each threat level and the proportion of species within that group that had seeds that were desiccation sensitive. And I said, as I mentioned, desiccation sensitive seeds have gotten the lion's share of research out of all exceptional species. But that means that they couldn't be dried and stored in seed banks as the global strategy target was hoping for. So if you look at the highest 3 threat levels on this graph, the critically endangered, endangered, and vulnerable, you can see that fewer than 75% of the species had seeds that could tolerate seed bank drying, and that it really was going to be impossible to meet the 75% target with conventional seed banking alone. So if conventional seed banking won't work and living collections will likely not be able to accommodate the number of species effectively, what are the other options for exceptional species conservation? They primarily center around in vitro and cryopreservation technologies, and that's what I'll be talking about next. In vitro refers to plant tissue culture. And tissue culture is a clonal method that most often refers to what we call micropropagation, or the propagation of plants through the outgrowth of lateral buds in a sterile container with a nutrient medium. The process starts, you can sort of follow this in the diagram here. It starts with a shoot tip taken from a plant growing in the wild. We surface sterilize that tissue and place it on a sterile nutrient medium that can stimulate the outgrowth of the lateral buds. These can then be separated, and each new lateral bud or shoot, can be placed on fresh medium, and to repeat the process and increase the number of shoots from that plant indefinitely. So that's sort of the circular diagram there in the middle. These shoots can be used as a source of shoot tips. And if you remember, the shoot tip of the plant has the meristem, the stem cells of the plant. Those can be used for cryopreservation. They can be stored and then thawed. And when they're thawed out, the shoot tips are put back onto medium to grow out into shoots, reestablish the culture, and the cycle can be continued. At some point, the shoots can be taken and put on a different medium to initiate roots. And once you have roots and shoots, you have a plant, and you can take it out and put it into soil, and you've got your plant recovered. I'll mention also that there's a second method that's very similar to this, that is somatic embryogenesis. That is, if you take the plant shoot tip or a leaf or some other tissue, put it in culture with a sort of a different medium and set of hormones. With some species, you can get clonal embryos to regenerate from that tissue. But they go through pretty much this same process. The somatic embryos can be grown and cultured indefinitely. They can be used for cryopreservation. They can also then be germinated, and plants can be recovered from them. So they fit very much the same into this same scheme that we have here. So both of those are very important in terms of producing plants clonally, and also for producing tissues for cryopreservation. Cryopreservation refers to the storage of tissues in liquid nitrogen, which is at -196 degrees centigrade. Sometimes people store things in the vapor phase at -135 degrees centigrade. Either one works pretty much the same. The tissues have to be protected from water damage, as we saw before, And either they're protected by drying if they have some natural desiccation tolerance, or they can be protected by cryoprotectant solutions, which actually do sort of a chemical drying to the tissues, withdrawing water and replacing water in the tissues so that when they're frozen, they don't go through that lethal ice stage, but they actually form a glass in the tissues. This process of cryopreservation can be used for a variety of tissues. You can see some of them here, and it really expands the types of tissues that we can store beyond seeds into somatic embryo cultures, shoot tips, as I've mentioned, also dormant buds of cold hardy woody plants, isolated embryo axes, pollen, gametophytes of bryophytes and ferns, and also fern spores and bryophyte spores. So just a wide range of tissues that are available. I should also mention algae. There's quite a bit of work on cryopreservation of algae as well. But it can really extend the life of these tissues for years or decades, and is the basis of really the kind of cryobanking that's needed for exceptional plants. Well, how do we apply these technologies? Well, this is the same diagram you saw before, but with a few other bells and whistles on it. You don't have to look at it really carefully, but I'll just direct your attention to each of the steps and how in vitro or cryopreservation methods can be used to overcome the challenge at that step. Starting with step 4, which is the seeds that might be deeply dormant and hard to recover from from seed banking, I should say. In vitro methods may be very useful here. As I mentioned, a lot of holly species show this deep dormancy because the embryos are immature when the seeds are shed, and it might take 2 or 3 years for the seeds to germinate. What's been shown with holly is that the embryos can be removed and germinated in vitro. This is a paper by Yang et al. From 2015, and you can sort of see the steps from A to B to C. The immature embryo is removed, and then they start to germinate in B, and you can see some leaf formation in C, and so on. Because there are over 100 threatened species of holly, this might be a very useful tool in recovering materials from seed banking. We move on to exceptionality factor 3, which is the freeze-sensitive or short-lived seeds. With many of these, this may be one of the easier approaches that is used, because many of these seeds can just be, instead of putting them at -20, you can put them in liquid nitrogen. And so the preparation for that and the recovery, the germination after that, are pretty much the same as seed banking, but you do need a cryo facility to actually do the storage of those seeds. And actually at the Millennium Seed Bank these days, they are, for species they suspect might be short-lived, they are automatically putting a portion of them into liquid nitrogen as well as into the -20. So there will be a lot of good data to follow with that. This is just an example of short-lived seeds from our lab, from a project a few years ago. We had some poplar seeds, Populus deltoides, stored for 20 years at 4 degrees and -20 degrees at the normal seed bank temperature and in liquid nitrogen. You can see from the graph, the bars, the first 2 bars show the germination of the fresh seed, and then the initial exposure to liquid nitrogen of the dried seed. We always do a test right at the beginning to show that the seeds can survive liquid nitrogen. And then they were put into storage at these 4 temperatures. After 20 years, there was no survival at 4 degrees, there was no survival at -20 degrees, but there was survival in liquid nitrogen. You can also see that the survival had decreased over the 20-year period, even in liquid nitrogen. Although a later analysis of this work, of this particular study, showed that the seeds that had been treated really in an ideal state at the beginning of the project and before they were put in liquid nitrogen, didn't lose much viability during that time. And it was seeds that had been maybe left out or put in the refrigerator for a couple of weeks before they were frozen. Those tended to decline even before they were stored, and so they continued to decline a little bit in liquid nitrogen. But in either case, liquid nitrogen extended the viability of those seeds much longer than the other methods could have. If we look at the exceptionality factor 2, you have seeds that are desiccation sensitive. And one of the methods that is used for those seeds is to remove the zygotic embryo from the seed and dry it and freeze it. Sometimes that will work with seeds that are, even if the whole seed is desiccation sensitive. I'll give you sort of a mixed example here just to illustrate it. As I mentioned, oaks have recalcitrant seeds, and people have used this method on some of the cold hardy oaks, and it has shown promise. But you can sort of see what, in the center picture here, that the axis is a very small portion of the seed that has to be removed, and it's dried and frozen. And then when it's recovered, it has to be recovered in vitro, because you've taken all the food away from that little embryo and you have to give it some nourishment for recovery. The actual pictures here, the other pictures I hear that I have are of Juglans nigra, which is actually the embryo system that we stored for 23 years in liquid nitrogen. It's since been shown that Juglans nigra is actually short-lived, not totally recalcitrant species, but the system is the same that we used. And just using it as an illustration. You can see we had very good survival after 23 years in liquid nitrogen. There was no loss in viability from the table here. We did have some embryos stored at -20, and they all were dead by the time 20 years came. But you can see this is the way the embryo is recovered in vitro in a test tube on a medium. And this is the black walnut embryo germinating after 23 years. Finally, we'll get to an area that we work a lot in these days, and that is in exceptionality factor 1, which is where seeds are not available, or there just are not very many seeds available for seed banking. And we go and actually initiate tissues and cryopreserve the tissues. So one example of that is Ziziphus salata, which is known as the Florida Ziziphus or Florida jujube. It's found in a very small portion of South Florida, or central South Florida. And it is growing in just a few isolated populations. The genetics of these populations is very depauperate, and so there's not much crossing that's possible between the plants in these populations, so they're just not producing seed. So just to illustrate how this process works, we receive shoots from our collaborators in Florida. They're usually sent to us overnight. We take off most of the leaves and cut it down to just the tip of about 1 inch or so in size, and surface sterilize that. And we do that with a dilute solution of commercial bleach, essentially. And then put the shoots into a sterile nutrient medium for germination, or for growth, I should say, not germination. And what hopefully will eventually happen is that we'll get a culture that looks something like that, that's producing nice shoots. And We would then go to the very tip of those shoots and dissect out the tiny shoot tip of that. It's about a millimeter in size that we need for cryopreservation. And it's sort of a meticulous process. You have to do it under a dissecting microscope, and it has to be done sterilely and all of that. But it does work, and it seems to work quite well with Sisyphus in the lines that we've been able to cryopreserve so far. So this is a method that is being used by us and by others to overcome that very first challenge if seeds are just not available for the banking process. Well, another question that comes up is cryobanking very effective? And we studied this in a project that was funded by IMLS in our lab a few years back, where we removed a variety of tissues of different types that had been stored in our cryobiobank for up to 20 years or 23 years in some cases. And I've mentioned a couple of these already. It included our short-lived seeds of poplar, which I mentioned. We also had a number of species of fern spores that germinated quite well after many years in liquid nitrogen. We had our zygotic embryos illustrated by the black walnut. We also had pollen frozen for 20 some years. It was American chestnut pollen, and it was viable after that time. And we had our postdoc, Danny de Ballesteros, who's shown down in the picture here. He took the pollen down to the American Chestnut Foundation farm in Virginia and pollinated trees, and they set seed and produced nuts. And so we were very excited about that. We also have frozen gametophytes of ferns and mosses, and those recovered quite well. And we also froze a number of shoot tips of a variety of endangered and non-endangered species of seed plants. And this is a list of the major plants that we cryopreserved as shoot tips. And you can see from the list here, they were frozen from anywhere from 4 to 19 years in cryo storage, and had a range of survivals. We had numerous genotypes of each one cryopreserved. And so we had a variety of recovery numbers that came out of this. But you can see that we had pretty good survival in many of these species. 40% is sort of the benchmark that people want to hit for this. And so after this amount of time, this was quite encouraging. There were 3 in red up here that you can see were somewhat low. Their initial survivals were a bit lower as well. But interestingly, while they were in storage, cryopreservation methods sort of evolved, and we have a better method now that came around. It's called droplet vitrification, which wasn't used in the initial bankings, and it seems to work better in many cases. So we're hoping that as we rebank a lot of these, we'll get better survival in the long term, because we're getting better survival in the short term with these. This just shows you a little bit an illustrator, a picture of a plate of Quercus virginiana shoot tips that are surviving after liquid nitrogen storage. Not long-term storage. This is just a 1 hour in liquid nitrogen, which is our experimental time that we use to test things out. But you can see, if you look at this plate, the very tiny little dots here are the original shoot tips. I just wanted to show this to illustrate how small the shoot tips are. It's 6 centimeters across on this plate. And those are about 1 millimeter or so in size. But you can see 3 of them have germinated well, or are resprouting, I guess you'd say, well after cryo, which was very encouraging for us. Oaks have been a focus of some of our attention lately, and they're somewhat more difficult than some other species. Well, so within the time frame that we and others have been able to study it, cryobanking has proven to be pretty adaptable to a wide range of tissues and many species. And it should be able to fill in the gaps that are seen with living collections and seed banks, and really really should be able to extend ex situ conservation to all species. All species haven't been tested yet, but I think the outlook is really good for cryopreservation to be able to preserve tissues of most any kinds of species. However, there's a lot of research that needs to be done to get to that point. I'll end up by talking a little bit about a few examples from our lab that can illustrate a little bit more about what we're doing and what exceptional plant research needs to, how it needs to move forward. Todson's pennyroyal, I mentioned before, is a mint. It's found in two population groups from the mountains of New Mexico. One of these populations was found on White Sands Missile Range, and we received funding from the US Army to do a genetic analysis of the populations. They were sort of interested in this because, as I said, the plant is not producing seed anymore. We don't really know why that is. It may have lost its pollinator, and it's reproducing clonally quite a bit. But the analysis, the genetic analysis, interestingly showed that there still is a fair amount of genetic diversity among the existing populations. But as a sort of a side project, because this plant wasn't producing seed, we took the opportunity to initiate in vitro cultures from the materials that were collected and to bank the shoot tips from those in vitro cultures in our cryo-biobank. So as a result of that project, we have 82 unique genotypes in our bank, representing 8 populations from distributed among these 2 larger population groups. We have 254 vials and over 2,500 shoot tips in cryo. I think we probably could have done more if we had funding directly for our project, but it was sort of a side project. And we enlisted students and interns and such to help with it. It sort of illustrates too that labor is a big part of this work. And it's one of the limiting factors in terms of banking materials in this way. I mentioned that we are starting to focus on oaks. All of them appear to be exceptional in the sense that their seeds cannot be stored conventionally. In the US, about 1/3 of them are threatened. And it's likely that about that number are threatened globally from the indications from the IUCN estimates. But they're sort of interesting because there are several options for biobanking for these species. As I mentioned, embryo axes have been tried and have worked for some of the species, but it may be that that method will work best for cold hardy oaks and not for many of the other ones. Somatic embryos have been initiated from oaks, particularly by some Spanish labs, and they have successfully cryopreserved those, which is exciting. And then last year we came out with a paper where we had demonstrated that shoot tips of oak cultures could also be cryopreserved. And we did this with Quercus virginiana, as I showed, the live oak, Quercus suber, the cork oak, and Quercus hinklei, the Hinckley's oak, which is the critically endangered oak I mentioned earlier. So shoot tip cryopreservation and somatic embryogenesis, somatic embryo cryopreservation, both appear to be very likely adaptable to a wide range of oak species. But I think there's two other methods that haven't really been tried yet, and that is cryopreserving dormant buds, which might be workable with species from cold hardy areas and cold hardy species, and then also pollen. It's been shown that pollen of many species can be cryopreserved, and I don't think it's been done yet with oaks, but there's talk that it should be, and I think that will help add to the conservation of oak genetic diversity ex situ. We're currently working with a small grant from the International Oak Society to continue this work on cryo. And we've also just recently received a small grant from Botanic Gardens Conservation International to sort of extend this work to some cycads to test out whether some of these methods might be workable with those as well. So finally, I'll mention a little bit more about our in vitro collections. I've talked mostly about in vitro as a way of getting materials for cryopreservation, but in vitro collections themselves can be very valuable. They're very labor intensive to maintain because every month or two you need to go in and reculture the tissues because they grow up and use the medium and that sort of thing. But they can be very useful for research, but also for producing plants that can be used for restoration projects. And we've been involved in that area a bit as well. And this is a list of some of the major projects that we've been involved with in Florida, Utah, Kentucky, with several species. The picture in the lower right-hand corner is a picture of a restoration project in Utah for the autumn buttercup, to which we produced and provided several hundred species for a couple different attempts at restoration. And it was interesting because each time they planted out, they learned a little bit more. We lost some plants, but having cultures that you can keep growing and then keep producing plants from is one benefit of using in vitro materials for restoration projects. So I think that is my last slide, and I will thank you very much for your attention. I will thank the folks listed here who have contributed to the work in the plant lab over the last few years in these projects. And I will look forward to questions if you have any. Thank you.
All right. Thank you so much. That was really fantastic, really interesting, important work. We've got a question from Bob about sort of space constraints and storing embryos, seeds. Are you running out of space in the facility that you're based in now, or are these samples small enough that you could potentially store sort of almost infinite numbers of replicates of species?
Well, that's a good question. We aren't running out of space yet. We have tanks. I think there were a couple of pictures of them there. And each tank can hold, well, it depends on the tank, but maybe from a few hundred to a couple thousand vials. And the vials are about this big. And they, each vial, we usually have about, if it's shoot tips, about 10 shoot tips in a vial. We could fit more in, but we want to space them out so they're not all in one place. If you have seeds or pollen or spores, you can have thousands of those in a vial. So space is at the moment isn't a big problem, partly because we're not a huge operation. I'd like to get to the point where we had, we were seams with these things, but yeah, it's interesting. But I think that's one of the advantages is that you can store a lot in a small space.
Yeah. So we also had a question that sort of gets at sort of plant physiology, and Lorraine was asking if you can explain a bit more about how our northern oak species make it through our winters given that they show some level of intolerance to freezing in seed banks.
Yeah,
I guess, I mean, some of the oaks have acorns on their branches through a winter. I guess they're not drying down in that situation, or
they may not be quite as dry, but I think, yeah, I had always thought it was that they got buried by animals
yes,
and
yeah,
protected that way. I'm not an expert on that, but I think a lot of the ones that survive are underground.
yeah.
Yeah.
Yeah. So another question, this is something I was wondering about as well. Karen's asking how the seed banks and cryo facilities like you've been describing are protected from extreme weather events, power outages, hurricanes, that sort of thing, given the time spans you're interested in preserving these for over, decades or conceivably, centuries at a sort of societal level. Are these how long can you go out without power, for example, and not risk losing the collections in one of these facilities? Is the liquid nitrogen, stable for weeks inside these tanks, or?
Yeah. Yeah, it sort of depends what you're doing. If it's liquid nitrogen, it's quite, these tanks are pretty stable. And as I mentioned, even the vapor will keep the
Mhm.
material okay for a while. We have ours in liquid nitrogen, so if we had to wait a while, it would be fine for quite a long time. The liquid nitrogen does not evaporate from these very quickly.
Mhm.
So we top them off every week and only need to add maybe an inch or so. And the tank is a couple of feet high. And we have small tanks, and a lot of places have huge
Yeah.
ones. If you're in a place where the conventional seed banks do have backup generators, for example,
Mhm.
but that is an issue. If you are a small seed bank and are using a chest freezer or something, which a lot of botanical gardens are, um, you run that risk. But they're well insulated and can last for a while.
Yeah. So we had a question from Lisa here about your organization at the Cincinnati Zoo. You described a lot of really fascinating experiments and these sort of long-term studies looking at viability after 20 years. And she was wondering what may be the sort of the broader goal of your organization in terms of, the number of species that you would like to see preserved in your collection, or sort of the diversity of species, or there's particular groups that you're focusing on?
Yeah, I think earlier on, our research was directed primarily by our collaborations with other botanical gardens. We had a couple grants from the Institute of Museum and Library Services to work with the Center for Plant Conservation Botanic Gardens, and they nominated species that they thought needed either in vitro work or cryo work or both, that would be good candidates. And so those were the ones we got started working with. More recently, we have, as I said, started focusing on groups like oaks,
Yeah.
because they're important in many ways. They're high profile, they are exceptional, and they fit our mission quite well.
Great. And there's an interesting question here from Karen about sort of question if there's genetic instability or the potential for mutations to build up over time when you're doing tissue culture and sort of going through multiple cycles of buds and back and buds through those cycles that you were describing.
Yes, that is a possibility. And that's one reason why it's nice if you can get the material into cryopreservation
Mhm.
sooner rather than later. Um, I think the genetic variations can occur, but the data on that is very variable depending on the species and the conditions. So I'm not quite as worried about that as maybe maybe it sounds like to me, it's not as big a problem as it may sound that it
Yeah.
could be, but I think it's something we want to watch. And we've actually looked at some of that with some of our groups and seen some of the genetic changes, but it also depends on the methods that you use for genetic analysis, what you
Mhm.
see, and how granular it gets, and what does that actually mean. So it's a big open question at the moment, I think.
Yeah. And I actually had a question that's sort of related to that about um, storing some of these species long term. So you're collecting some rare endangered species and moving it into sort of deep freeze and preserving that genotype that was collected 5 years ago, 10 years ago. Um, what is the thinking about the issue of sort of microevolution in those wild populations, and sort of you're preserving this genotype that might have been adapted to conditions 10 years ago, 20 years ago, but with rapid climate change, you've sort of frozen that species genetics at some point in time. So for restoration efforts 50 years in the future, is there thought about sort of the lack of microevolution that would happen in that species while it's sort of sitting on ice in a seed bank or a cryo bank?
Yeah, I think that's going to be a challenge either with conventional seed banking or cryobanking. It would be the same. As things change, uh, what we're trying to do is keep a backup
Mhm.
if you lose something in the wild. But I think if that does happen and you bring the material out, you're going to have to look at it and say, is it going to go back to where it was, or do we put it somewhere else?
Yeah. Okay. Interesting. And we have one other question here from George asking just about the challenges, difficulties of thawing out these embryos or the seeds, and sort of what's involved there. Is it as simple as bringing them out and setting them on a bench top and letting them thaw out, or is there a very meticulous stepwise sort of warming up? How do they come out of this deep freeze?
Yeah, that's a good question. It sort of depends on the method that you use to do the freezing. If you're freezing something like a seed, let's say a short-lived seed that's just been dried, as you would normally put it into a seed bank, but you put it into cryo, um, probably you just put it out on a bench top and let it
Mhm.
thaw, and then you germinate it. For these other kinds of tissues that we're doing, where we're going through all these cryoprotectant steps and things to get them into culture or to cryo, you do need to do some thawing steps. It's not too complicated, but
Mhm.
um, basically putting it in, thawing it very quickly. And
Okay.
with the method we're using right now, you just take the tissues that are in a very, very tiny bit of frozen cryoprotectant, uh, quickly put them into a room temperature solution, and they immediately thaw out. So the rate of thawing is very important in some, depending on the tissues that you're using, but the faster the better usually for those. And it's the same for freezing. The faster you freeze these things, the
Mhm.
better it is. Mhm.
All right. I had one other question, and folks feel free to add more comments or questions into the chat here. In one of the initial graphs you were showing, you were describing this distinction between the formation of ice crystals, but then if you dry the seed, the moisture content of the seed down, and then chill it, you get formation of sort of glass. And what specifically is that terminology meaning about glass? Like, what are those glass-like characteristics? And is that's the tissues and the cells taking on those characteristics?
Well, I'm not a biophysicist, but
Yeah.
it's um, technically an amorphous solid.
Mhm.
And my understanding is the cytoplasm is sort of, you can think of it as solidified in place, essentially,
Yeah. So
rather
no crystal
than,
formation.
right,
Yeah.
no crystals.
Okay. Okay. Yeah, no, that's interesting. Um, all right. Well, thank you again for this talk. I think it's really fascinating and very topical, given the threats to biodiversity and so many exceptional species out there. And we've had a talk last winter from Wes Knapp talking about endemic species and extinctions in those groups. And so this is sort of a wonderful sort of counterpart to that, thinking about how some of these species might be preserved in seed banks or cryobanks. So we really appreciate you coming out tonight and sharing some of this research. And the amount of work you've done over so many years is just amazing. So thank you so much.
So thank you for having me.
Yes. All right.